Okay, is that showing up? Okay, excellent. Yeah, well, thanks for that really lovely intro. Indeed, before I started my life as a biology researcher, I taught a little bit of high school biology in Ecuador, and I think is the most biology I've ever learned in a year was actually teaching it at the 10th grade level. Yeah, but thanks again for the invite, and it's really lovely to be here with you guys today. So again, my name is James Crall. I'm going to tell you today about some pretty recent series of work trying to understand the effects of pesticide exposure in bees, in particular in the context of the sort of complex array of social behaviors in bee colonies. And if you came here looking for info on honeybees, my apologies, that was false advertising. You'll see nothing, but you will learn about their fascinating cousins, the bumblebees. So that's primarily what I'm going to tell you about today. So I just want to start off by reminding you probably of things you already know, which is that in addition to being just a fascinating group of organisms, bees are incredibly important for both conservation and food production. So the vast majority of flowering plants on the planet are in some way dependent on animal pollinators. In addition, about a third of our global food system, which I think is a primary interest for the course here, comes from pollinator dependent crops. And in existing systems, we think that about a quarter of existing yield gaps. So if you look at within a particular crop, the difference between high and low yielding farms and try to understand what drives that difference in yield gap, about a quarter of that might be due to lack of sufficient pollination. So this is an important real world problem with important impacts on food production. And in addition to just managed commercial pollinators like honeybees and also some bumblebees, we know that pollinator diversity, increasingly we're learning, is really important for food production. And in addition to just the sort of economic impacts of food production, pollinators are incredibly important for the health of humans, primarily because the crops that are pollinator dependent, so that third of the food system is especially rich in micronutrients. It's vegetables, nuts, and fruits that are very micronutrient rich. And so there's been some really nice work recently trying to sort of model out what would be the health impacts of, in this case, complete pollinator loss. But we would anticipate that the changes in the food system associated with loss of pollinators would have really substantial impacts on human health across the globe. And so with that in mind about the sort of importance of bees for both conservation and food production, it's worth noting that we are, of course, changing the environment in many, many different ways that impact the health of pollinators. So one of those that we'll talk a lot about today is the addition of insecticides and other agrochemicals into the environment. So introducing novel stressors into the world of bees. In addition, we're of course changing landscapes in ways that change the habitat availability, nesting resources, and food resources for bees and other insects. By moving animals, and in particular pollinators around, we're introducing novel disease risk. And of course, we are fundamentally changing the abiotic conditions on the planet through climate change in ways that we know impact pollinators and bees. So lots of different ways in which we're changing the environment. An additional challenge in understanding how these changes might affect bees in the food system is that these stressors don't act independently, right? So they interact and can have synergistic impacts that alter the performance and life of pollinators, both at the individual scale, so they can kind of have direct physiological impacts on individuals, but also in the case of the sort of unique biological case of social pollinators, which are especially important. We also want to understand not just those direct impacts on individuals, but how those scale up to whole group level impacts, and also how that social context might feed back to and potentially modulate individual effects of these stressors. So within that big picture of the ways in which we're changing the environment and their impacts on pollinators, what I'm going to focus on today is really the impacts of insecticides. And in particular, I'm going to focus on neonicotinoid pesticides. You are likely familiar with these, but just as a reminder, these are a relatively novel class of insecticides that target nicotinic acetylcholine receptors in the insect central nervous system. And so that's really targeting the primary excitatory neurotransmission system in the central nervous system of insects. And this is a somewhat dated at this point graph, but of their showing the explosion in use of these compounds of a few different particular chemicals in this class of neonicotinoids over the course of about 2 decades since their introduction in the US. And so now they are quite widespread, the single most commonly used class of insecticides across the globe. And one of the challenges about them is they are quite persistent in the environment, and they're because of that very widespread. And so, for example, one of the common ways in which they are applied is through coating of seeds that get the compounds that get taken up systemically within the plant and then show up in all the plant tissues, which is great if you're a grower that wants resistance in all of your tissues throughout the course of your plant's life cycle. But it also means they show up in nectar and pollen produced by that plant, which is one of the primary routes of exposure to bees. And so to just give you a sense of the scale of that exposure, this is a absolutely delicious sounding study where this group ordered just as much honey as they could from as many places they could across the globe. So sounds absolutely lovely until you learn what they actually did, which was then went and tested all those global samples of honey for neonicotinoid pesticides. And the white dots you'll see here are the samples that did not contain detectable neonicotinoid. You will notice that those are the minority. So roughly 3/4 of the samples of honey across the globe contain detectable neonicotinoid. And so this is honeybees going out sampling flowers and telling you that they are being directly exposed to these compounds. And often this was multiple exposure. So kind of the most frequent mode is not just being exposed to one of these neonicotinoids, but multiple simultaneously. And there's really now pretty compelling evidence that even low levels of what we would say is sublethal exposure to these compounds nonetheless can impair colony growth. And we'll get a little more into what I mean by sublethal in a moment, but essentially this means exposure below directly toxic levels can nonetheless impact sort of colony success and many aspects of phenotypic performance in bees and other insects. And in addition, there's pretty compelling evidence that this can have sort of trophic cascading effects. So for example, in aquatic ecosystems, wiping out the invertebrate population in lakes that can then impact fisheries and sort of have scaled up ecosystem effects. Okay, so if we sort of focus on trying to think about the impacts of these compounds on bees and trying to understand what they do, there's kind of a few big challenges I want to focus on today. And this is really even just focusing on the relatively narrow set of effects that are the behavioral impacts of pesticides. And the reason I'm kind of focusing on that today is we suspect that a lot of the important, as a neurotoxin, right, it's likely that at least some of the important effects are behavioral. And these are all below the sublethal or below the lethal level in the sense that they might not be directly toxic effect where a bee exposed to a particular concentration is dead within 48 hours, but nonetheless, there might be subtle alterations in behavior that lead to long-term impairment of colony growth and therefore populations. So if we want to think about that genre of impacts, there are kind of a few big challenges, I'd say. So one is, it's pretty complicated just to understand even the direct effects of a single agrochemical, right? Or multiple agrochemicals, because, you know, bees live an incredibly diverse behavioral life, right? They do a lot of different things in a lot of different contexts. And so we want to understand what any particular chemical does to not just a single behavior, but kind of the complex suite of behaviors, right? And also that's true for all of the enormous variety of neuroactive pesticides that we're putting into the environment that can have quite different effects because of different toxic pharmacology and sort of chemical level effects. So that's sort of one challenge is just understanding the sort of direct behavioral effects across this behavioral suite. But again, as I sort of mentioned before, these effects are likely not isolated in the sense that just looking at these sort of direct behavioral effects of a single pesticide might not give you a sense of how that patterns of multiple exposure impact bees, or how exposure to a particular insecticide interacts with other things like environmental conditions, nutritional stress, the many other sort of relevant aspects of stressor in the life of a bee that living outside, right? So we want to understand not just those direct effects, but how they interact with other stressors. And finally, the sort of third tier of this challenge, I think, is that it's likely, and there's good reason to think, that these effects can also vary even within the life cycle of a single species or across different species. So, there's about 20,000 species of bees across the globe, and that's just bees, not even talking about other insects and pollinators, much less the 5 million species of insects across the globe. Just in North America, we have about 4,000 native pollinators, and they all have their own sort of unique set of behaviors they have to perform. So we want to understand not just the direct effects, but also how they interact with other stressors, and then how those interactions and complexities might vary across the life cycle of a bee and across species. So sounds like a pretty daunting challenge, right? But what I want to try and do today is really just give you some examples of how we can work through some of these complexities, really focusing on a particular example that I think is a nice species for thinking about some of these challenges. So today what I'm going to do is first start by kind of introducing you to my favorite pollinator, which is bumblebees, a group of about 200 species of, in North America, really our most important native pollinators. So I'll give you a little introduction to start. Then, paralleling the sort of challenge that I just laid out, I'll tell you about some of the direct impacts of neonicotinoid exposure on bumblebees, then focus on some of the interactive effects and the way we've studied that, and finally finish by looking at sort of differential effects of these compounds across development. Okay, so to start with just a little introduction to the beautiful bumblebee. So this is a pretty typical day in the life of a bumblebee, and I'll just play that again, highlighting something you might have missed, which is a single bumblebee worker maneuvering through this sort of remarkably complex environment. And this is a pretty normal scene in some sense. We all get used to this, especially if you chase pollinators. This is a scene you get used to, right? But this is quite a remarkable moment, right? This animal is maneuvering through this spatially complex environment that is also, temporally dynamic. It's sort of like maneuvering a helicopter through buildings that are swaying back and forth, right? It's a real challenge, all with a brain that has, orders of magnitude fewer neurons than a human brain, packs all its energy on board, and dramatically outperforms any sort of human-made drones, right? So really kind of a remarkable biological phenomenon, just maneuvering through this environment. And that worker is out trying to get food resources, so pollen and nectar, to bring back to the colony. And this is just sort of showing you an example. This is a high-speed video of just a bumblebee navigating in a wind tunnel to a food source. And you can see these sort of a little, just an example of that, these exquisite, subtle changes in the movements of wings that drive the ability to maneuver through that environment. So, but all of that life that we see of pollinators outside, gathering food from flowers, is really actually aimed at something that is often more hidden from us. So this is, you'll see a bumblebee worker return to a little hole in the ground. And what she is doing is actually bringing food back to an underground nest. This is a common eastern bumblebee, Bombus impatiens, and bringing that food back to fuel the development of the nest, which is all happening underground in the case of bumblebees, right? So this is a look inside of a bumblebee colony. You can see workers moving around, taking care of developing young, fueling all this growth of the colony within the nest. And bumblebees do this in some quite challenging and quite diverse environment. So this is a bumblebee queen out foraging and trying to start a nest on top of the Continental Divide at about 12,000 feet in the Rocky Mountains. And this is, they're cold weather and challenging environment specialists. They live from the tropics to Alaska, and so inhabit this sort of incredible variety of environments. And in contrast to honeybees, bumblebees have a pretty unique and interesting life cycle. So unlike honeybees that overwinter with, often tens of thousands of workers, they actually start life off as a single solitary queen. And so in the northern hemisphere, in say April, you'll see these queens out, these really big bumblebees out foraging on flowers, and those are actually the queens starting a new nest. They're doing their own foraging and bringing back resources to the nest to lay eggs. Those eggs will eventually become workers that take up some of the work in the colony. So they start doing foraging and caring for developing young within the colony. And at least, here in Boston, from say April to around August, September is when you see this really strong period of colony growth. And then eventually the colony toward the end of the growing season sends off new reproductives. So this is the whole colony's life cycle is really aimed at producing new males and queens. They will leave the colony, mate, and then everyone except for the queen dies. Basically, there are no males or no workers over winter. That mated queen will save sperm from usually a single mating, find a nice hole in the ground to sleep in, wait out winter to start that cycle again in the spring. So really sort of fascinating life history of the colony here. So to break into and kind of study this system, what's really nice about bumblebees as a study system is they come in a box from the internet. So they've been commercialized largely for greenhouse pollination, especially. And there are certain crop species that need especially buzz pollination, which is a unique kind of pollination. Tomatoes require this, where the flower has to be actively buzzed to release pollen, and bumblebees can do that, but honeybees can't. So for a variety of reasons, they've been commercialized. So you can order this great little cardboard box of bees on the internet, and then you can do things like connect that hive to the outside world and let that colony, in this case, fly in and out of a little obstacle course where we can do things like monitor flight performance and get really a sort of holistic view of the colony performance. So for example, with this system, we can look inside of the nest and monitor nest behavior, foraging activity, flight performance, all under sort of naturally varying environmental conditions. I'm not going to tell you too much about today. Feel free to ask me in the Q&A, but I'm not going to tell you much about what's happening outside of the wall here. It's fun, but it's somewhat less relevant. But what I do want to tell you a little bit is how we study the behavior of bees within the colony and their sort of foraging activity. So this is an example of a bumblebee worker coming out through a little foraging tunnel where they're connected to the outside world. And you might notice some very strange markings on the back of this bee, not natural variation. It turns out this is a little paper tag that carries a barcode that lets us uniquely track and monitor individual workers within a colony. And so we can use a computer to basically go back and identify who this worker is and also where she's going. And so use this to sort of continuously monitor, in this case, foraging activity as bees are coming in and out. And that gives us a really nice view into how, not only what the whole colony level activity is, but how it varies across individuals. We can also use that same tracking approach to then look inside of the colony and monitor what bees are doing as they're sort of fueling that growth and all that activity within the nest. And so this is a view again inside of a Bombus impatiens, common eastern bumblebee colony. You can see it has a lot of the same stuff as a honeybee colony. So in the upper left there, you can see some pots filled with nectar. More toward the middle, these sort of yellow splotchy balls that are developing brood. And so it has a lot of the same stuff as in honeybee colonies. You just don't get the same sort of sacred geometry that you get in. So it's the slightly less organized cousins maybe of honeybees. But now we can take that same view now through a tracking camera and actually use that same approach to monitor the movements, interactions, behavior, and identity of uniquely identified workers across the bumblebee colony. So this gives us a really nice view into the behavioral life of a whole bee colony, right? And we've learned some really interesting things that I'm going to sort of breeze through quickly. But one of the striking things that we learn by looking inside of these colonies is there's just an enormous amount of individual behavioral variation that is somewhat hidden from us. So for example, if we use that system combined with foraging, monitoring to sort of track the behavioral life of a single individual, this is 2 days in the life of a single worker up here on top with different behavioral states sort of colored in here. So red is when this bee was foraging, and with the different colors sort of showing different behaviors within the nest, correlating with where they are and how active they are. So you can see this is a bee that spent most of her days foraging. But if we repeat that same thing across here, 7 workers, you see really striking patterns of individual variation that is quite stable from day to day. So most workers kind of dabble in all behaviors, but they have a really strong tendency toward particular behaviors that is stable over time. Another really striking correlated pattern is that there's very strong patterns of individual spatial occupancy within the nest. And bear with me a little bit, this is going down in a rabbit hole, but it will actually come back a little bit. So I do want to just kind of highlight this a little bit. So if we take all that tracking data within a colony and sort of create a little home range, a map of where that bee spends time within the nest, here this graph is showing a sort of heat map of that, where this bee kind of like to be in the middle right. This is associated with where the developing brood, the developing young are within the colony. And we can see that bee's spatial pattern is pretty consistent across days, right? If we look at that same pattern across individuals, we see lots of variation in where bees spend time that is very stable across days. And without digging too much into this, one thing we found is that, in fact, that variation in where bees spend time actually is really important in driving how the colony responds to sort of changing labor demands. So if, for example, you force a bunch of bees to forage, where they are within the nest will determine how they switch to foraging. So all that was really just to give you a sense of a little introduction to the life of a bumblebee colony and some sense of how we study it. Okay, so with that in mind, now I want to shift gears a little bit to focus on how exposure to neonicotinoids in particular affects these behaviors. And in particular, I'm going to focus on, so I would say there's a relatively better understanding of how low levels of exposure impact behaviors like foraging outside of the nest and things like navigation and the ability to sort of gather resources and bring them back to the colony. But what's been a little bit more of a black box, both figuratively and also literally, because it happens in a hole in the ground, is how exposure to neonicotinoids impacts this sort of important suite of behaviors within colonies that drive colony growth. So that's really what I'm going to kind of focus on today. So what happens within colonies to workers within the nest that we know were exposed to neonicotinoids? So to study this, we can, for example, remove individual workers from the colony. And this is a single worker being dosed with a single sort of dollop of imidacloprid, the single most common neonic used globally. And so what we can do is use that same tracking approach we used before and now remove individual workers from the colony after getting baseline behavioral data for every worker, and then randomly assign them to treatment groups. So for example, some workers we just remove, feed a sucrose solution, and then put back in the colony, in part because, being ripped out of your nice, dark, warm, cozy nest and being, deprived of food for a bit, fed, and then put back in your colony might be pretty disruptive. So we want to know if there's any kind of baseline effects of just this removal and tagging. And then we also give some bees a low dose of imidacloprid and then a high dose. And I just want to highlight that even both of these doses are in what we think is within the realistic total exposure range. So these are sort of calibrated to be the total dose of imidacloprid we would expect a forager to consume in either a single foraging bout at the low dose, or in a single day at the high dose. So both sort of in the realistic range, we think. Okay, so now what we can do is look at changes in behavior within individual workers because we have data on what they're doing before and what they're doing after. So what I'm going to show you is changes in behavior across those different groups. So for example, if we look at changes in the amount of time spent nursing for our control group, good news, we don't see any obvious effect of just the treatment of being removed and put back. So on average, bees are basically doing the same thing when we put them back. No effect on nursing at our low dose. But at our high dose of imidacloprid, we do see a significant reduction in nursing activity. We see a similar pattern for overall activity. So at the high dose of imidacloprid, they're just spending less time moving around. There's also a spatial shift, so they're further out from the nest center. And then the other thing we can do is use just a proximity network to estimate social interactions within the colony. So basically measuring when bees bump into each other. And what we find is that our high dose bees have significantly fewer social interactions than our control and low dose bees. Okay, so all of that is sort of in this acute exposure paradigm, right? Where we remove bees, give them a dollop of neonic, and then put them back in the colony. But that's potentially unrealistic in a couple ways, right? So one, it's probably physiologically unrealistic in the sense of, even if it's a realistic dose in total, it might not be the natural pattern of exposure. So instead of sort of continuous low doses, which is how we expect the bees to be exposed in, say, a seed treated crop, they're getting just a single dollop, right? The other thing that it misses is actually the social structure of exposure, right? So we know that bees aren't randomly exposed to these pesticides. There's likely a social structure, and we might be sort of missing some of these social feedbacks in that acute exposure. So what we really want to do is then give colonies these sort of continuous low levels of exposure to really understand in a more realistic way what's happening in the field. The challenge with that is if you want to capture that natural, especially social structure, you can't just focus on individuals anymore. You need to focus on the colony as your sort of functional replicate, right? Which statistically becomes quite challenging if you need to monitor dozens or hundreds of individuals in each colony. This is sort of a classically challenging problem in social insects, right? So to get around this, what we did is built a robotic system that can automate that behavioral tracking and data capture across multiple colonies in parallel. So this is just a schematic of sort of benchtop system in the lab where we can program cameras, an array of cameras to go over the top of 12 different colonies sort of in succession. Each one of those has a sort of mini version of an outside and inside world, a lit chamber and a dark chamber for sort of outside foraging and the nest. And then within each one of those, we can again do the same kind of tracking I just showed you. And this is just what this looks like in the lab, with each one of those being a single colony. And this is the robotic platform sort of in action, with the cameras coming into focus over top of a single colony and starting to record data. And this is now just a time lapse of that system. So we sort of let this go continuously. We get about 12 trials a day for each colony, these sort of quick 5-minute trials. We can monitor 12 colonies in parallel. And this then sort of gets us up to the level of replication where we can start doing whole colony replicates as our sort of paradigm. So let's just get into this chronic exposure, more realistic patterns of exposure. So what do we see now if we do this sort of long-term low level of exposure? And what I'm going to show you are trials comparing just a sucrose exposed colony to a colony exposed to 6 parts per billion imidacloprid, which is sort of on the upper end of the realistic range of continuous concentrations that bees might be exposed to in nectar. And so this is just 2 sample colonies, sort of right at the moment of the beginning of a trial. So no sort of obvious difference, different colors there are just showing patterns of individual movement of bees. And this is those same colonies 72 hours later. On the bottom right, that is actually a video. You can see the one bee sort of floating around. So this is a pretty striking effect, right? Very clear reductions in activity. And so you might be thinking to yourself, like, very cool that you built a very fancy robot, but like, do you really need a robot to, to tell you about this or any fancy tracking, right? But what I'll show you in a second is that this effect has actually been somewhat hidden from us in some surprising ways. So let me tell you what I mean by that. So now what I'm going to show you is sort of composite data across multiple colonies for about a week of these trials. So here, every individual dot is the average activity. So just the average amount of time spent moving in a single colony averaged across their whole trial. And I'm going to then give a running average of, in this case, the green is going to show the control colonies. And so what we see is overall a pretty high level of activity with a bit of a circadian rhythm, which you expect. And so the gray bars here are showing overnight periods of this sort of continuous data collection for about a week. And so you see some reduction in activity overnight, which is more or less what we expect because there's some bees that are sort of continuously active. And actually nurses have reduced circadian rhythms compared to foragers. So this is more or less what we expect to see at the aggregate scale. So now if we compare that to imidacloprid exposed colonies, you see a really interesting pattern. So you see an overall reduction in activity, but the effect has a circadian rhythm. So the strongest effects are actually occurring overnight. And by the end of our trials, you see no detectable difference during the day in activity, but still a really significant effect overnight. So that colony I was just showing you that looks just totally wiped out was a trial from overnight. And that colony to eye actually looks fairly normal during the day. And so you see these sort of strong activity crashes overnight. And it turns out we see similar patterns with other metrics. So if we look at the amount of time spent nursing, again, we see if we compare the control and imidacloprid exposed colonies during the day, you see a little bit of a trend, but no significant difference or no significant effect of our treatment during the day, but a significant reduction in nursing overnight. If we look at, again, that's the sort of spatial pattern we saw in acute exposure. CBs are further out from the nest center or trending in that direction during the day, but no significant effect, but then a significant spatial shift overnight. And if we look at now sort of an aggregate metric of that social interaction pattern, so this is just of all possible interactions that could happen within a colony, how many of them actually happened, no reduction during the day, significantly at least, and a significant reduction in social interactions overnight. So I think the takeaway from these chronic exposure trials is we see overall sort of qualitatively similar effects to acute exposure, but that those patterns vary with the time of day. So they're primarily happening overnight, or they're at least stronger overnight than during the day. And also, didn't really present this, but we've also done some modeling to suggest that those multiple effects, so shifts in activity, shifts in nursing, spatial shifts, and social interactions are probably functionally linked. So if you tweak one of those behavioral knobs, you're likely to get out the other effects as well. So for example, if you just alter the sensitivity of bees to physical contact with nestmates, you actually get them spending more time on the periphery of the nest and doing less work in the center of the nest. So it's probably best to think of these as multiple outcomes of a potentially like sort of single direct physiological effect of these compounds. Okay, so that gives us a sense of some of the complex ways, in addition to sort of known effects on foraging, tells us there are likely some significant effects on this sort of other suite of behaviors that we maybe didn't know so much about before. So now I want to shift gears a little bit and think about in addition to sort of direct effects, what happens if we think about the effects of these compounds in the context of a second environmental variable. And then what I want to think about is really temperature, because it's particularly relevant for bumblebees and other bees. So bumblebees, like honeybees, do this really kind of amazing group behavior where individual body temperatures fluctuate, and we think of them as, cold-blooded insects, right? But actually the whole colony runs a body temperature, and they will maintain the temperature of developing brood within a quite narrow range. And they do this even in the face of sort of fluctuating temperatures in the external environment. We think this is important for their ability to colonize these kind of cold, challenging environments and continue, and also be able to emerge early in spring, and allow larval growth to happen efficiently, even in these challenging temperature environments. So this is, again, the sort of tracking camera, and on the right, a thermal camera view of a colony experiencing a cold stress, and so you can see them sort of clustering around the developing brood and generating heat. And so what we can do is study this behavior in outdoor conditions. These are some commercial bumblebee colonies that we just stick temperature probes directly in contact with developing brood and sort of just measure how well bees are doing this important behavior. So here, this is in gray, showing the outdoor air temperature over the course of a little more than a week. And you can see in a control colony, they're doing a pretty good job. There's some fluctuation, but in general, they're keeping that temperature within sort of a few degree band of optimal growth. But now if we compare this to an imidacloprid exposed colony, we see much more substantial fluctuations in the brood temperature. So an impaired ability to sort of homeostatically regulate that temperature within a narrow range. So indeed, it looks like it does impair social thermoregulation in ways that might make them more sensitive to temperature fluctuations. And we see this effect consistently across colonies. And importantly, we see it happening much stronger. So if you sort of looked at just thermoregulation around, say, 30 degrees Celsius air temperature, you wouldn't see much of an effect of this treatment. But as temperatures start getting colder, that effect becomes more apparent. So it's stronger in colder temperatures. And also, it's dynamic. So I'm not showing you this here, but it looks like the particularly strong effects are on the ability of a colony to recover from cold temperatures. So when sort of temperatures are rising, that's when we see the strongest effects. Okay, so that tells you a little bit about this sort of active brood thermoregulation, but there's another tool in the thermoregulatory toolkit of bumblebees, which is the ability to sort of change nest architecture. So I'll show you what I mean by that. This is a colony, sort of a commercial colony coming right out of the box, where you can see in some colonies, after a few days, they'll start building up a sort of wax covering, which you can see on the bottom here. And in some cases, it gets really extreme and actually envelops the whole colony. And the idea is this is essentially an insulating layer, right? We think it basically works like a nice warm blanket, gives you more passive insulation in the face of cold temperatures. So if we compare our control and imidacloprid colonies in the conditions they were in, almost all of our control colonies built some amount of this wax covering, and none of our imidacloprid colonies did. So likely exposure also impairs the ability to sort of actively restructure the nest. Okay, so that tells us that in addition to sort of direct effects in nice, clean laboratory conditions, there are also likely interactive effects with a stressor like temperature in naturalistic outdoor conditions. The last thing I want to do, I don't know where I'm at on time, but yeah, okay, so I'll try and spend just a about 5 minutes telling you a little bit about the sort of most recent work we've been doing, trying to look at how these effects might vary with colony development. So differential effects across this colony life cycle. So why might this be important? So this is some data from a field study that was done looking at the sort of in realistic field conditions where some fields are treated with seed-coated neonicotinoid and some aren't. What are the impacts in these fully realistic field conditions on bees of different types? So what they found is if you look at solitary bee density or nesting, you see strong reductions in the abundance of wild bees in these fields that are treated with neonicotinoids. And as a reminder, or if you don't know it, the vast majority of these species are actually solitary, right? So the best known are the social ones, but the vast majority live sort of standard insect lives alone most of the year, doing their own solitary foraging. If we look at bumblebees in these fields, we do see strong reductions in sort of colony growth and strength. But if you look at honeybees in the same fields, you don't see any detectable effect of the same exposure, seemingly, right? So there could be a lot of things explaining that pattern. Honeybees are the most sort of maintained, most bred of all these species, and much more even than the commercial bumblebees. But also there's a really strong social gradient here, right? So the solitary bees obviously are solitary. Bumblebees usually have colonies of up to 100 or a few hundred individuals, whereas honeybees live in colonies of tens of thousands of workers, right? So it could be that something about living in that very big colony might buffer some of the effects of pesticides, right? And bumblebees are a really interesting system to think about this question, because as I sort of introduced before, they actually have a social gradient within a single colony's life cycle, right? So they start out as a solitary individual and then become an integrated social colony. So can we use that pattern of development and that variation in colony size to think about this question a little bit, and whether or not colony size and development drive differential sensitivity? And we've also done a little bit of modeling, which I won't dig into the details, but if we sort of make a very dumb version of life inside of a bumblebee colony where you treat them as dots moving around and then play in some of the known effects of pesticides and then artificially vary in simulation the size of the colony, we can look at the difference between imidacloprid exposed and control workers in this sort of simulated world. And here on the right, you're seeing a graph of the results of that where the color here is showing you the difference between the two groups, imidacloprid exposed and control bees, with darker colors showing less difference between those. So you can think of that as the strength of effect of imidacloprid. And the pattern you should see is that in larger colony sizes, we see reduced effects of imidacloprid exposure in this sort of simulated world. So some empirical evidence and some sort of theoretical reasons to think that there might be a social gradient in pesticide or stress sensitivity. So to get at this experimentally, we take advantage of the fact that you can bring in wild bumblebee queens and develop them in the lab. So we do this with lots of colonies in parallel and then let them develop from a single queen to a fully mature colony. And then with random timing, we pull them out and give them a 2-day stress treatment. That is a combination of some colonies just receive a control, some receive 10 parts per billion imidacloprid in sucrose again, some receive just a cold exposure, and then some receive imidacloprid and cold. So randomly assigned treatment during development with random timing with respect to colony development and size. And we can make a modified version of that tracking chamber where we now make it look space age with insulation on the outside and control temperature within colonies. And this is an example of a colony experiencing one of these transient cold stresses where for a few hours we drop temperature within colonies, and you can see them sort of activity dropping a little bit, clustering toward the center of the nest, and also lots of active heat production directly on the brood. So if we sort of quantify the behavioral responses, one thing we can do is measure that clustering by just taking the distance between bees. And you can see as it gets colder, they do get closer together. So we can sort of measure that behavioral response. And it turns out if you take all the suite of things we can measure within a colony and take sort of a single metric that tries to capture as many of those variables as possible, called a principal component, the most important one seems to have to do with spatial structuring, in parallel to what I was telling you about before about sort of spatial variation in terms of workers, where workers are within a colony. And what we see is this sort of principal component of behavior. Higher values of it mean more proximity to the brood, more interactions with nestmates, and lots of other sort of variables associated with this clustering response. And we do see that in cold exposure, we see that the value of that metric go up. So it looks like this is a good metric of sort of behavioral response to cold. And so now what we can do is sort of test this idea that the effects of these stressors are stronger in small colonies. So what I'm going to show you is these are effect plots. So on the left here, we're looking at the sort of mean difference between sucrose and imidacloprid exposed colonies that were also exposed to cold on the left here. And then on the y-axis is the sort of aggregate score on that centrality metric. So what we see is what we expect. In control colonies, we see a relatively high centrality score, so that when they're exposed to cold, so that means they are sort of clustering. If we look at the same effect in the absence of cold, we don't see much effect. Okay, so this is telling you in small colonies, this is what we more or less expect to see. But now if we look at the effect across colony size, the important thing to see here is on the left, you see that gradient flattening out, which means there's less of an effect of these combined exposures in small colonies than in larger colonies. So this is what we expect under this hypothesis that small colonies might be more sensitive. Sorry, I'm gonna blow through a little bit of the end of here, but I can come back to this in questions. We see a similar pattern for eventual effects on long-term colony growth. So we see stronger behavioral effects in small colonies, and we also see that paralleled in sort of long-term effects on how well the colonies do. Okay, so just sort of in conclusion, on that overall, I think what these results tell us is, indeed, neonicotinoid exposure does impair important aspects of social behavior within colonies, but they're complex, they vary over time. In addition to these just isolated direct effects, there are interactive effects with secondary stressors like temperature. And also that we have evidence that those effects vary over the colony cycle, right? So it looks like these small young colonies are more sensitive to these stressors than larger, more mature colonies. Okay, so now just zooming out again to these kind of grand challenges and understanding the effects of pesticides. So how do we think about the direct phenotypic effects? They are diverse. They're amongst these many behaviors that bees do, they're likely many of them are affected by these exposures, and we want to understand that diversity. We also find evidence for strong and complex synergies between single exposures and additional stressors like temperature, and also evidence that in this case, at least colony size and development do drive sensitivity to these stressors, which could have implications for both the sort of timing of exposure. It might mean that in bumblebees, April early season exposure is more important than July or August exposure, and also sort of supports this idea that solitary bees may be more sensitive than social bees. In terms of sort of broad conclusions, I think we can draw from this might seem a little bit depressing in terms of our ability to understand the effects of these compounds that we're putting out into the world. Because one sort of big takeaway is it tells us that often we study these compounds sort of in the middle of the day in nice warm laboratory conditions and in mature colonies. And a practical takeaway is I think that might be a sort of list of the worst case scenario for thinking about the real world effects of these compounds. But also I think there is real promise in these sort of scalable techniques that are really just getting easier and better to use to understand this sort of broad suite of behavioral impacts of these compounds and potentially might be helpful for developing better testing paradigms and understanding the real world effects of the many chemicals that we are introducing into the world of bees and other insects. Okay, and with that, I'll just say that this is the work of many folks and many generous funders. And if we have time, which may not, I'd be happy to take a question, but thanks again.
If anyone who's not registered for the class has a question that they want to ask James, now is your opportunity to do that. And we'll just take one because we're running a little bit behind on time. All right, well, we've got one question, so we'll go ahead and ask it. It was submitted by Christina. So in honeybees, foragers have a stronger circadian rhythm than nurses. Was there individual variation in circadian rhythm in the bumblebees in response to the insecticide?
Yeah, that's a really great question. So it looks like, short answer to that is we don't find super obvious, if you try to divide by sort of foragers and nurses, there's not a super obvious effect in our system, but I don't actually think that's good evidence that there isn't differential effects, in part because we might be seeing slightly weaker circadian effects because they're not doing natural foraging. So I think it would be really cool to do the parallel in fully free foraging that might help you get at that a little better. But yeah, no obvious evidence that we do see differential effects, but great question.
All right, looks like we have one more, and I feel like we should definitely take this one. It's from Richard Rouge. Given the wide use of insecticides and their perceived value to farmers, how can we best protect bees?
Well, nice simple one to end the, the lecture. I mean, I think maybe the shortest way to sum up my thinking on this question is that we need to, so I think there are some obvious effects, right? So obviously we need to better understand what the real world effects on the pollinators are and balance that against the needs for real pest management, right? I think there are some cases where it looks like that might be sort of an easy-ish solution, in the sense that there might be cases where actually there's not a huge measurable benefit to using imidacloprid or other neonicotinoids or other pest controls, and they're having negative effects on pollinators, but actually not having that much measurable benefit to farmers, right? So there might be some simple cases like that. And I think the broader answer is really trying to think about all this in this, I think the IPPM framework is the right one, right? So thinking about how you not necessarily wholly eliminate or wholly use neonicotinoids, but how you integrate it in the sort of complex, more measured and flexible decision-making structure, which is probably not a particularly satisfying answer, in part because it's a complex problem. But I think, the first step is really understanding what the impacts are and what the real world effects on pollinators and what that means for farmers, and sort of quantifying that, I think is really important as sort of a first step. And then we can think about the challenging balance.
All right, well, with that, I think we'll conclude our session for the week. Thank you for everybody who was willing to attend and participate. And thank you, James, for giving your lecture today. And thank you to Fabia, Stacy, and Teresa for introducing the speaker this week. So we'll take everybody into our private discussion room and continue the session. For those of you not joining us, thank you, and we'll see you again next week.
Thanks all.